So let's move on to another session for today, Phytosome and Advanced Drug Delivery System. We have with us an eminent speaker, Mr. Prakash Khandagale, sir. Mr. Prakash Khandagale, sir, is an assistant professor at St. John Institute of Pharmacy and Research. Sir graduated in pharmacy in 2009 and completed his master's in 2011 from Pune University. He has around 5 years of academic experience. Sir has published several papers. He is a life member of Maharashtra State Pharmacy Council. With this brief introduction, I would now request Sir to take over the session.
Thank you for the nice introduction. So we will continue with the next session, the phytosomes. Phytosomes, novel drug delivery system. Okay, so we will proceed. So novel drug delivery system with respect to specifically the herbals. Now the question should raise in mind why the herbal should be preferred for this the novel drug delivery system. If you are knowing the advantages of the different herbs, okay, for example, the easy availability of the herbal phytoconstant, the herbal drugs, okay, the less side effects, even the drugs that can be easily administered. Okay. Besides this, some of the advantages, there are also some disadvantages with respect to the herbal drugs. Most of the drugs that are taken without the prescription, so in that conditions, there might be chances of some of the allergic reactions to a particular individual. Okay. So besides this, still this herbal drugs, they are offering some advantages to formulating the different formulation using this, the novel drug delivery technology, simply we can say. So for a longer time, if you're saying this, the herbal drugs, they were not considered for this herbal drug technology or this novel herbal drug technology because of some the scientific justification that is needed for any particular drug, or we can say some process requirement with respect to any standardization in extraction techniques. Okay. So moreover, now the modern phytopharmaceutical research, they can solve these aspects related to this whatever the scientific needs. For example, any the site of action, any mechanism of action of particular drug, the drug reduce concentrations with the help of using the different novel drug delivery system. So some of this novel drug delivery system that has been mentioned in the slide, you can see over here. The liposomal drug delivery system, the next one, that phytosomal one, the microspheres, okay, which is the latest, that one easily ingested one. Microemulsion system, the that transfersomal drug delivery system, even the you can say this is non-invasive the type of the drug delivery system. Okay, even that ethosomal drug delivery system and some solid lipid nanoparticle systems. The solid lipid nanoparticle system that has attracted the much attention with respect to the this novel drug delivery system with using the herbal drugs. Now, with respect to the introduction of what is this phytosome technology. Now, if you see in the word phytosome, it's the combination of two different words. The phyto, that simply stands for the plant, and the some word, that is a cell-like. So this phytosome is one of, we can simply say that the vesicular novel drug delivery technology, wherein the phytoconstants that will bind with the phospholipid carrier that we will see, which is the phospholipid carrier as we proceed further. So this is the brief one with respect to this phytosome. Here mainly involve that chemical interaction between the phospholipid and the water-soluble herbal compounds, specifically the hydrophilic compounds. And these are mainly suitable as the drug delivery system for the topical and the oral administrations. So if you're seeing this phytosome technology, that similar somewhat to your the liposomes, okay, the liposome technology, and mainly this the phytosome that are prepared through the attachment of the individual ingredients of whatever the herbal extracts that are going to extract the different phytoconstituents based on the nature of the solubility of the phytoconstituents in a particular solvents. Okay. So to your any phospholipid carrier, that is here in our case that phosphatidylcholine, that is species that resulting in a formulation that having the higher solubility and hence the better absorption, that leading to your promoted pharmacokinetic and the pharmacodynamic properties. If you see that pharmacokinetic, that with respect to your right from the absorption of the drug, okay, that distribution, that metabolism and excretion, the pharmacodynamics. So pharmacodynamics, simply we can say the study of the biochemical aspects and the physiological effect of that particular drug. Okay. So that this phytosome technology that offers several advantages compared to the conventional herbal extract. If you're seeing that the conventional herbal extract, they're having the lot of the disadvantages, so there are some limitations. So limitations mainly if you're seeing with respect to the instability with the in acidic pH, then the degradation, you want the pre-systemic metabolism in the liver, the stability concern, the absorption concern. So most of these limitations that can be overcome with using this phytosome technology. It can offer the different advantages with respect to the increasing that the bioavailability, which is a very important factor. Then the reduced drug concentration even also. The next one, that is the phospholipid carrier. As I said to you that one of the phospholipid carrier that is mainly used in case of this phytosome technology, that is the phosphatidylcholine molecules. So this phosphatidylcholine, if you see in your structure, that mainly the phosphate group and that the choline molecules. Here the choline moiety that mainly interact with combining with the phytoconstituents of the herbal extract, and this the phosphate group that simply envelopes, okay, this the choline phytoconstituent, so that will forms the phytophosphatidylcholine complex, and that will give the major effect with respect to the herbal formulations. Now, the properties of the phytosomes. Now, properties with respect to some physico-chemical properties or some biochemical that biological properties. If you see in the very first one, the phytosomes that they can accommodate the active principle that simply anchor to the polar head of the phospholipid group, that becoming an integral part of the membrane. So most of that the hydrophilic phytoconstituents, they they simply experience that the limitations while crossing your lipid membrane layer. Okay, so that can be overcome here with the help of this anchoring the polar head of your phospholipid, the choline molecules. Second thing, that the interaction between the phospholipid and the substrate is due to mainly the development of the hydrogen bond. Okay. So here the hydrogen bond formation takes place between the polar head of the phospholipid molecule and the polar functionalities of the chief phytoconstituents, and that enable to cross your lipid bilayer of the biological membranes. And this once that cross that lipid layer, that they can betterly absorb and that are utilized, and as a result that they produce the better results or simply the high therapeutic index. Then the phytosomes that are the lipophilic substances with a definite melting point that are freely soluble in case of the non-polar solvents, for example, your toluene solvents, and that moderately soluble in fats. Some of them are unstable in alcohol solvents. Okay. So when treated with water, this phytosomal complex, they assume a mycelial shape that forming the structures that mainly resembles the liposomes, and that exhibiting this fundamental differences between these two. We see that the fundamental difference between these two, that is what is the difference between the phytosomes and the liposome technology. Our next point, that is the mechanism of the phytosome technology. Now, as I said to you that mainly here that interaction that mainly takes place between the phosphate group, the choline moiety, and the hydrophilic nature of your phytoconstituents. So mainly this phytosome that results from the reaction or that reaction of that stoichiometric amount of the definite proportion of, simply we can say, the phospholipid molecule, that is phosphatidylcholine, with the standardized extract or the polyphenolic constituents, simple like simply or the flavonoids. So if you see in that the polyphenolic compounds or most of the phytoconstituents like take your the glycosides, the tannins even also, they're having the large molecular size, okay, and they're feeling that constant with respect to the absorption or in turn simply the bioavailability of that particular phytoconstituent. So that can be overcome with the help of with your this phytosome technology. So here the phosphatidylcholine, which is our the carrier, okay. Beside that, acting as a carrier for the drug itself, it nourishes your skin organs. It's a bifunctional compound, that phosphatidyl moiety that being lipophilic in nature, and that the choline moiety that being the hydrophilic in nature that we were seeing in the previous slides, that structure of your the phosphatidyl choline. And basically here that the choline head of your the specie molecule that binds to this phytoconstituents, while the lipid soluble phosphatidyl portion that comprising the body and the tail, which simply envelopes your the choline bound material, and that forms your, we can say that the phosphatidyl complex. So the preparation of phytosome. Next point, that is preparation of phytosome. Simply, this is a general mechanism for the formation of your the phytosome suspension. So in this one, you have to take the phospholipids, simply dissolve in any organic solvent containing your the drug or extract. Then the solution of phospholipids you will get in that particular drug. Simply allow it to dry with the help of the spray drying or the tray drying. Then formation of the thin film. Then next stage that only the hydration that will form the micellar solutions. Okay, and in that result, basically in the formation of your the phytosome suspensions. So this is the generalized mechanism for the preparation of your the phytosome. Then next one that we will see some of the examples with respect to the preparation of this phytosome. So very first one, take the soilocene phospholipid that are simply reacted with any polyphenolic extract in the equal ratio with 5 ml of your the DCM solution with stirring until it get evaporated. And once your this DCM solvent that was evaporated, to that add the 5 ml of the N-hexane solution to the thin film with continuous stirring and left simply in the film hood for the complete removal of the solvent. So after complete removal of that particular N-hexane, the thin film that was hydrated, okay, and the next is that is the sonication, that sonicated for in order to form the desired phytosomal complex. Now second example, that is the naringine, that naringine phosphatidylcholine phytosomes. So it's also one of that the flavonoid compounds. So naringine PC complex that was mainly prepared by taking your naringine with an equimolar concentrations of your the phospholipid. Generally in this phytosome technology, you have to take your the drug and your phospholipid carrier in 1:1 ratio or 2:1 ratio depending upon the substance. So here the equimolar concentration of your the PC, that is phosphatidylcholine, and that naringine we are placing 100 ml of RBF, and that reflects in the dichloromethane for 3 hours. So on concentrating that solution for 5 to 10 ml initially that you are taking, so greater volume of your the solvent, so you have to concentrate that solution to about the 5 to 10 ml. To that add the 30 ml of your the N-hexane that was added in order to get the complex as a precipitate. So after getting precipitate, simply go for the filtration. So collect that precipitate and place in the vacuum desiccator. A third example, that is with respect to your the silavine phospholipid complex preparation. Okay. So here again that take the required amount of your the drug, that is silavine, and take your phospholipid carrier that simply place in 100 ml of your the RBF and dissolve in an address ethanol solution. Okay. So after ethanol that was evaporated off under the vacuum at 50 degrees Celsius, the dried residue that simply gathered and placed in the desiccator overnight, and then crush in the mortar and simply sealed with your the 100 mesh size, and that resultant that silavine PC complex that was simply transferred into your the glass bottle, and you have to flush with the inert gas, simply here the nitrogen, and simply store in the room temperature. Now, next important point, the selection of the phytoconstituents, so the formulation considerations. So what are the factors that mainly we can say to some extent that limits the formation of any particular the complex formation with any the phospholipid carrier? So the three different factors in this one, the very first one, that is the selection of the herbal extracts. So herbal extract, as we are knowing that the mixture of your the different phytoconstants, and they possess the different properties. So properties such as the antioxidant, that moisturizing, astringent, anti-irritant, then your hepatoprotection or that the photoprotections. So because of the each unique properties, they simply produce the healing effect, the softening effect, the rejuvenating or the sunscreen effect on the skin, and that simply improve your the pharmacological and that pharmacokinetic profile in the body. Okay. So initially that you have to carry out the literature survey of that particular herbs. You have to correlate that activity of any herbal compound based on that any particular category of the phytoquestions. For example, whether there is a presence of any flavonoid compounds or monoterpene compounds, any polyphenolic compounds, or the tannins, or that endocannabinoid compounds, or organosulfides. Okay, so based on that, one can select that herbal extract on the basis of their nature, availability. So availability of that herbs is very important thing than the estimation method, the stability and the utility of the developed formulation, as well as any reported previous research on that particular herb or that particular phytoconstants. That is the very important thing. The next one, that is the nature of phytoconstants. So in this one, solubility is an important criteria. So based on that nature of your the phytoconstant, the solubility in particular solvents, that whether it is a hydrophilic or lipophilic, the best suitable formulation that can be selected. Now, third point, that is the selection of your the dosage form for delivery of phytosomes. Now, the dosage form selection, that is the very important one. So it's mainly based on its potential for improving that effectiveness and simply the efficiency of that particular bioactive compounds. So our purpose is simply to improve its efficacy regarding that continuous action of the herbs and the systemic effect on the human body. And different properties of that herbal drugs, some inherent properties of herbal drugs like your the hydrophilic nature or that hydrophobic nature, even the surface characteristics of that the system, such as any permeability factor or the charges. Okay. So degree of biodegradability and the tonicity, even that release profile, okay, and the size of the product required of the formulation that need to be taken into consideration. So these are all that factor that needs to be taken into consideration while formulating this phytosome technology with respect to any of the herbal drugs or that phytoconstants. So the next one, we will see what are the different dosage forms. So some of the suggested dosage forms for the phytosome delivery that has been mentioned over here. So here the soft gelatin capsule, then hard gelatin capsules, some of the tablets that can be also prepared. Okay. So very first one, that the soft gelatin capsule. So here in this, the phytosome that simply that can be dispersed in any oily vehicles, that any vegetable source or any semi-synthetic source in order to obtain that suspensions which can be fill your the soft gelatin capsules. Second one, that is the hard gelatin capsules. So this phytosomes that can be filled into your hard gelatin capsules. So here to keep in mind the direct volumetric filling process without the pre-compression. So pre-compression might affect to some extent the disintegration time. So you have to take care of that things. Without pre-compression, that the direct volumetric filling can be applied in case of this hard gelatin capsule formation. Even the low density of your the phytosome complex that seems to limit the maximum amount of powder that can be filled into the capsules, and it has been found that up to the 300 milligram of that for that size capsules, the powder can be filled in that one. So with the use of the piston pump capsule filling process, we can increase the amount of the powder to be filled in the capsule. So next one, that is the tablets. So in the tablets, very important thing, the weight granulation that should be avoided. So due to some negative effect of your the water and even that heat effect on the stability of your the phytos or that phytophospholipid complex, it has been found near about up to only 37 to 40 degrees Celsius that it can tolerate that maximum temperatures, and due to the limited flowability, even the stickiness and the low apparent density of your the phytosomal complex, here the direct compression process that can be applied only for the lower doses or the lower unitary doses, and should be diluted with your the 60 to 70% of the excipients, not to obtain the tablets with the desired characteristics. The next one, that is the topical dosage form. So as we have seen in the introduction, this phytosome that can be a very good, that we can say the option for even the oral or topical dosage forms. So firstly, in this one, you have to prepare the emulsion at the lower temperature, not higher than the 40 degrees Celsius. And after that, incorporates that phytosome complex into it, and that resultant phytophospholipid complexes that are simply dispersible in the main lipid solvents which are employed in your the topical formulations. Now, next point, that with respect to your this differentiating point between your this phytosome and the liposomes. So here in the diagram, you can completely see in here the drug molecule that mainly that encircle with this phospholipid the aggregates, the hundreds or thousands of the phospholipid aggregates that are simply that encircles your the drug molecules. Whereas in case of your this phytosomes, your drug molecules is completely enclosed within your this phospholipid carrier. So that will the great advantage for this using the phytosomal technology, and that can easily cross your lipid bilayer membrane, and that will result in your increase of bioavailability of that particular drug. So if you're seeing that the differentiating point specifically with respect to the concentration of the drug molecule and the phospholipid carrier, the phytosome simply the phospholipid and your plant phytoconstituents that actually forms your the 1s to 1 or 2s to 1 molecular complex depending upon the substance complex. Whereas in case of the liposome, here the liposome is mainly formed by mixing your the water soluble substance with the phosphatidylcholine which your phospholipid carrier in a definite ratio under the specific condition. As I said to you, there is no definite ratio in case of your the liposomes. Here the hundreds or thousands of your simply the phospholipid molecules there forms the aggregates with respect to the drugs. Second things, the bonding. Here the phytosomes that mainly involves that chemical interaction or the chemical bonding between your the drug and phospholipid carrier molecules. So in case of here the liposomes, here no chemical bond is formed, and that phosphatidylcholine molecules, okay, that simply surrounds your the water soluble substance. Third thing, with respect to that absorption, so the bioavailability simply we can say, so the phytosomes that are much better absorbed than your liposomes, that showing the better availability, whereas your liposomes, the bioavailability of liposome is less as compared to your the phytosomes. So here same that differentiating point that has been mentioned with respect to that the concentration of your the drug phytoconstituents and your the phospholipid carrier. Second thing, the bonding, and the third thing with respect to the bioavailability or that increased effect of your the particular technology. Some of the marketed phytosome preparation. So here you can see that some of the marketed phytosome preparation with respect to the particular plant phytoconstituents, their biological activity, or that applications of your phytosomal technology. So very good example of this one, that is the curcumin, okay, that anti-cancer or that antioxidant drug. And with the application of phytosomal technology, it has been found it has shown the improved on that antioxidant activity, and the most important thing that is your the bioavailability. The next one, that is the embelline, that the embelline that has an antibacterial activity, okay, that the solubility enhancement. Then comes your some of the drugs like your the naringine, even also the epigallocatechin, that anti-cancer antioxidant effect, that also found to be the increase in absorption enhancement, okay, the naringine compound, that anti-cancer anti-inflammatory compounds, okay, that also prolong that actions and that enhance bioavailability of that particular drug. Even some traditional drugs like with respect to your the ginsenosides, okay, which is the immunomodulator, okay, and the widely used nutraceutical product, it has been found there is an absorption that has been increased with the applications of your the phytosomal technology. Now some product that has been mentioned over here with respect to this the phytosome, you can see here the panaginson phytosome 150 milligram dose as a food product as applications. The green select, okay, that 50 to 100 milligram, that which is the rich in antioxidant compounds, the best choice for protection against your cancer and the damage to your cholesterol. Again, the leukoselect phytosomes, which is also having the systemic antioxidant or that specific activity, best choice for the most people under the age of the 50, and also specific for the eyes, your the lungs, the diabetes condition, the varicose veins, and that provides the protection against the heart disease. Okay. Then here the sabaselect phytosomes. So this sabaselect phytosome is one of the immunomodulator that enhances the immune function in response to your the toxic challenge. These are some of the marketed formulation you can see over here, that green tea phytosomes, then the milk is phytosomes. Very known that the curcumin, that bio curcumin phytosomes, the grape seed, okay, the hawthorn, and that the grape seed phytosome that has been mentioned over here. The next point, that is the evaluation of the phytosomes. Now, the evaluation of phytosomes, so yeah, the point that has been mentioned with respect to the visualization effect and the vesicle size or the zeta potential, the entrapment efficiency, the transition temperature, then the surface tension activity measurement, the vesicle stability, the drug content, and spectroscopic evaluation. So we will see all these evaluation parameters in details. So the drug entrapment and the loading capacity. So very important things. There is a no problem with respect to this the drug entrapment with respect to the phytosomal technology. So here the drug phytosomal complex that are mainly centrifuged at 10,000 rpm for 90 minutes at 4 degrees Celsius. You have to separate your phytosome from the untapped drugs. So the concentration of the free drug that can be measured by doing that your UV spectroscopy, and the percentage drug entrapment, the entrapment efficiency that can be calculated with the help of the formula you can see over here. The weight of the total drug minus your weight of the free drug divided by weight of the total drug. Next one, that is the visualization effect. So here the visualization of that phytosomes that can be achieved using the transmission electron microscopy, or by using the scanning electron microscope. So we'll see that the scanning electron microscope that's mainly used to determine the size of the particle and its appearance. So here the dry sample that was placed on your electron microscope brass, the brass tube which is coated with the gold ion in an anesthetic, and the random scanning of that complex is done as 100. Okay, that magnification. With respect to the transmission electron microscopy that mainly used to characterize the size of your phytosomal vesicles with 1000 magnification. So with the help of this one, we can see that the particle size determination or the visualization of the phytosomes. Next one, that is the transition temperature. So here the transition temperature of that vesicular lipid system. So as I said to you, this phytosome technology is one of that vesicular drug delivery technology. Okay. So here the transition temperature of that vesicular lipid system that can be determined by this differential scanning colorimeter. So different transition temperature that changes from one phase to the another, that can be determined with the help of this differential scanning colorimeter. The next one, that is surface tension activity measurement. So here the surface tension activity of the drug in the aqueous solution that can be measured with the help of the ring method in the Dunoy ring tensiometer, okay, which is the force or the weight to the liquid in that lamellar drug solutions. Okay, then the vesicle stability. Now the stability of the vesicles that can be determined by assessing the size and structure of vesicles over the period of time, and the mean size is measured by the DLS and the structural changes that are monitored by the transmission electron microscopy. Then the next one, that is the drug content. So the amount of the drug that can be quantified by with the help of your modified HPLC technology or by using any suitable that spectroscopic method. Now spectroscopic evaluation. So this is very important evaluation parameters in order to confirm the formation of the complex. So as I said to you that there is a formation of the complex between the phytoconstituents, and on the other hand there is your phospholipid carrier, which is that is phosphatidylcholine. So in order to confirm the formation of complex, or to study the reciprocal interaction between the phytoconstituent and the phospholipids, the spectroscopic methods are used, mainly the HNMR or the C13 NMR that are used. So here's some of the research studies that has been mentioned. So here the NMR spectra of that catechin and its stoichiometric complex with the phospholipid carrier, that is diasteryl phosphatidylcholine, that have been studied by the Bombardelli. And in non-polar solvent, there is a marked change of the HNMR signal that originating from the atoms which are involved in the formation of the complex, without any summation of that signal that is peculiar to your individual molecules, and whatever the signal that from the protons that belonging to simply the flavonoids that are to be broadened, than that the protein that cannot be relieved. And in this the phospholipid, there is a broadening of all the signals, while there is a singlet that mainly corresponding to the tertiary amine of the choline that undergoes an uplift shift, and the heating that sample at 60 degrees Celsius that mainly results in the appearance of some new broad bands, which corresponds mainly to your the resonance of the flavonoid moiety. Then next one, that's C13 NMR. So here, that's in case of this C13 NMR, again the same example with the catechin and its stoichiometric complex with your the diasteroyl phosphatidylcholine, that particularly when recorded in your the solvent at the room temperature, that all the flavonoid carbons that are clearly invisible one. So whatever the signals that corresponding to your the glycerol and that choline portion of the lipid, okay, that are broadened and some are shifted, while most of the resonances of your fatty acid chains, they retain their original sharp line shape. And after heating to 60 degrees Celsius, what all the signals belonging to your the flavonoid moieties, they reappear again. Okay, although that they are still very broad and partially overlapping. Then next one, that is FTR spectroscopy. So with the help of the FTR, the formation of the complex that can be confirmed by the iris spectroscopy simply by comparing the spectrum of the complex with the spectrum of the individual components and their mechanical mixtures. Okay. So this technology or this spectroscopy is simply the useful tool for the control of your the stability of the phytosomes when you are going to disperse or micro disperse in water or when incorporated in a very simple cosmetic gels. So from practical point of view, this stability that can be compared by comparing simply the spectrum of the complex in the solid form with respect to your phytosomes with the spectrum of its any micro dispersion in the water after that lyophilization at different times. So you have to simply compare that spectrum of your complex. Okay. The next one, that is the in vitro and that in vivo evaluations. Okay. So this evaluation that is simply selected on the basis of an expected therapeutic activity of the biologically active phytoconstituents which are present in your phytosomes. For example, in case of your in vitro anti-hepatotoxic activity. Okay. So this keep in mind this phospholipid carrier, that is your the phosphatidylcholine. Okay. That is also having hepatoprotective activity. Okay. So here the in vitro anti-hepatotoxic activity that can be simply assessed by the antioxidant and the free radical scavenging activity of your phytosomes. And for assessing the in vivo anti-hepatotoxic activity, the effect of the prepared phytosomes, okay, on the animals that can be studied against the different drugs like the thiazotamide, your paracetamol, or the alcohol-induced hepatotoxicity. Again, that the skin sensitizations and the tolerability studies of the glyceric acid phytosomes, the assignment, a commercial product that simply describe the in vivo set evaluation methodology with respect to this one. The next one, that is what are the advantages of the phytosomes. Okay. So phytosomes, as we have seen, the phospholipid, which is your carrier, is one of the valuable components of your phytosomes, which has a bifunctional activity by acting as a vehicle, as a carrier, as well as the health benefits. As acetu that itself has a hepatoprotective activity. So second thing, that absorption of that hydrophilic active constants is increased with the help of this technology, as your drug is totally entrapped, okay, within that the phospholipid carrier, which also increases that efficacy, and that resultant efficacy increases the dosage requirement is also reduced. Okay. Third advantage, that it has the better stability. Again, that the phytosome has the ability to permeate through the skin due to its the lipid layer around the phytoconstituents, and that will enhance the effectiveness. Next one, that by increasing the solubility of the bile to your the herbal origin phytoconstituents, this phytosome they mainly enhance the liver targeting. Also, the low risk profile with respect to this phytosome technology, as no large scale drug development risks, since the toxicological profiles of your the phytosomal components that are well documented in the scientific literatures, even that no costly equipment that is required for manufacturing of this phytosomal formulations. Besides these advantages, there is a very major disadvantage of this the phytosome technology. Sometimes the leaching of the phytoconstituents of the cell, that is the cell, which reduces the desired drug concentration, which is the major disadvantage of this phytosome technology. Now, the next point, that is what is the recent research on this with respect to the phytosome technology. So one of the recent research on the Salvium marinium. So this drug, the Salvium marinium, the milk thistle, which mainly contain that liver protecting flavonoid drugs, and that the three flavonoids of this one, the subclass flavonoids, that having the fully saturated the serines, and this silabine that mainly predominates, followed by the two different derivatives, that is siladenine and the silacristin. And this xylabine is actually a flavolignan compounds probably within the plant by combination of your this flavonol with your the coniferol alcohol, and which is the most important of these three different derivatives. And this xylabine has been shown to have positive effects in treating the liver diseases of the various kinds, including your the hepatitis, your cirrhosis, your the fatty infiltration of the liver, and even the inflammation of your the bile duct. The next one, that the antioxidant capacity of this one, the silymarin, the same the phytoquestion that substantially boost your the liver's resistance to the toxic chemicals. The fruits of your the milk thistle plant that mainly contains the flavonoid, and this milk thistle plant is flavonoid rich compounds. That flavonoid rich plant is known for its superlative effects, and the standardized extracts from this the Silabine marinum is an excellent liver protectant, but it's poorly absorbed orally. It has some also some limitations with respect to this, the oral absorption of with respect to this one. And this Silabine mainly protects your liver by conserving the glutathione in the parenchymal cells, while your this phospholipid carrier that helps to repair and replace your the cell membranes. And these constants that are likely to offer the synergistic effect of your the sparing the liver cells from the destructions. So this synergistic effect is also one of the advantage of your the phytosome technology. This synergistic effect of different cosmetics, okay, that will also helpful to overcome the different stressful conditions from the different exogenous or the endogenous factors. Okay. So in its native form within this milk thistle fruit, this silabine occurs primarily, which is complex with the sugars as a flavanol glycosides or the flavolignan compounds, and this has been extensively researched and found to have the impressive bioactivity and bioavailability, and also there's one limitation. This is limited by some poor bioavailability of these compounds. Next, the research of that telescope that reported that the silymarin phytosomes, they show the better anti-hepatotoxic activity than silymarin alone. Okay, and that can provide the protection against the toxic effects of your the aflatoxins B1 on the performance of that the broiler chicks. The next scientist that that they have carried out the one research. They prepared this silymarin phytosomes and studied its pharmacokinetics in the rats. And in the study, they have found that there is an increase remarkably after oral administration of that the prepared this drug of phospholipid complex due to an impressive improvement of the lipophilic property of your this complex, and there is an improvement of your the biological effect of this silymarin. So these are the references with respect to your this the phytosome and advanced drug delivery systems. Thank you all.
Good afternoon, everyone. It's my privilege to have been asked to propose a vote of thanks for today's session. I would like to thank our eminent speaker, Mr. Prakash Khandagale, sir, for enlightening us with his knowledge. Today's webinar gave us a deep insight into the topic phytosome and advanced drug delivery system. Sir has mentioned various points of like the properties of phytosomes, the mechanism, the preparation of phytosomes, what are the consideration for selecting a phytosome, what are the marketed preparations and the evaluations of phytosome. Our sincere thanks to Chairman Mr. Albert D'Souza, sir, for his encouragement, and thanks to all other dignitaries of the management. A special thanks to our principal, Dr. Savita Toro ma'am, for her immense support. I would also like to thank the coordinators for their guidance and support. The feedback link will be provided in your respective WhatsApp group. Kindly fill it. That will help us to improve our program. Thank you all for being there with us. Have a great day.